sperm through the vas deferens into the urethra is known as, which of these words? Emission. Okay? And let's go down on these questions here. Okay, the embryonic vas deferens develops from which of these structures? Mesonephric duct. Okay, the mesonephros is an intermediate kidney. It's the tube associated with it is the mesonephric duct. Paramesonephric, we'll see later today, is the female oviduct and uterus part of the vagina. All right, the erectile tissue, which forms the corpus spongiosum of the penis. Now, here's an example of what some practices I want you to try while you're taking the online quizzes and the lecture exam, is to focus on the question or question statement before you look at the answers. All right? Now, if you just read the erectile tissue, which forms the corpus spongiosum of the penis, there's no question there. All right? It's asking you to complete the sentence. So you obviously have to look at the answers to do so. But I want you to pause. You'll have plenty of time to take the exam. It's 2 hours, and most of you won't need all of that time. None of you need all that time. Most of you won't take all of that time. So stop before you look at the rest of the answers and say, all right, erectile tissue, corpus spongiosum. What is it? Where is it? Maybe draw a diagram. What becomes the corpus spongiosum? And think about what you know about that information. All right? Then look through the answers. That way you are attacking the question instead of being buffeted back and forth and starting to question your knowledge as you read each of those statements. Oh, I don't know this. I wasn't sure of that. I don't remember reading that. And you start becoming confused and upset, and then you're not sure. You think that's the answer, but you're not sure. All right? So some statements like this one, you can answer without looking at the suggestions. So cover it up. The embryonic vas deferens develops from. Think about it in your mind. Come up with the answer. And if you see it there, mark it. Okay? Something like this, still think about it. Even if you have to look at the answers to know what the actual question is, what do you know about the corpus spongiosum? Okay, it contains the urethra. It develops from the bulb. It's on the ventral surface. It develops into the glans penis. So you can think about these, maybe write little notes or draw the diagram. Then read the question. The rest of the answers, some suggestions. So it's innervated by the sympathetic nervous system. We haven't talked about that. So you don't know about that. Maybe you studied it and you don't remember, or you never looked at it. But you do know a lot about the corpus spongiosum. So let's keep reading. It's formed by the erectile tissue of the crus. What's it formed by? Develops from the bulb. Okay? Contains a segment of the urethra. You already thought about that before you looked at the rest of the suggestions, so you know that is the correct answer. You can just mark it. There's no A and B, C and D, all the above. You know that one is correct, even if you don't know anything about the other ones. Okay? It's compressed by the ischial-cavernosus muscle. You may remember that. You may know that that's not that one. It's the bulbospongiosis. But even if you don't, you figured this one out. Okay? That's what I mean by being in charge, seating yourself in what you know about the content of that question before you get confused by what you may or may not know about the answers. All right? Again, I will not make up words. All right? So I won't make up a nerve or make up the name of a muscle or some hormone. I mean, it's about something, you know, here and there, but it won't be on purpose to confuse you by moving a letter out of place. All right. Identify this gland. Prostate gland. Now, that would be a type of lab quiz or exam question without the answers. So we will have multiple choice lab questions at the very end. Your digestive system and the comprehensive optional lab exam will have multiple choice. All right. The peritoneum is pulled into the inguinal canal and scrotal sac in form. So without even thinking about looking at all those other words, peritoneum, is it the innermost, the outermost, the middle layer of the scrotal wall? Innermost. All right. As the testes descends, does it push it through or does it grab it and pull it down in front? Grabs it and pulls it down in front. Is it a muscle? No. So as we look through here, all right, hopefully you would catch the two What are these structures? Another type of lab question.
Different ductile.
Different ductals, okay? Yes. And then we have the reedy testes, straight tubules, seminiferous stuff.
Where's the straight?
Pardon?
Where are the straight?
The straights are where two seminiferous tubules join together. So each of these right here, the straight part of the seminiferous tubule would be the straight tubule. Some books call it the tubuli recti. Same thing, straight tubule.
Sorry, can you repeat that?
Which part?
The straight tubule?
The straight tubule. So when I drew it on the board on Tuesday, we had the highly curvaceous, well not curvaceous, highly coiled seminiferous tubules. Two of them join together to form a single straight tubule.
Okay.
So it's this structure right here, these structures here, just proximal to the reedy test. Okay? All right, so here's our inguinal canal that's been opened up. You can see this structure as the superficial ring. What do we call these muscle fibers that pass through the inguinal ring and are part of the spermatic cord covering? Premaster muscle, okay? Identify this cell. Ignore the letters that tell you what it is over here. All right, so spermatogonium here, here. These are what? Primary spermatocyte. That's what will be on your quiz next week. Okay, no histology on the male reproductive today. Just the organs and model structures. Okay? And these angular nuclei belong to late spermatid, while these small round ones up here are early. Now, even though this one is out of sequence, it still has the punctate or dotted appearance, so it's still a primary spermatocyte. All right, there'll be folds in the Sertoli cell, so we might find some early spermatids, you know, deeper in relationship to primary, if you want to go by their appearance. The only thing that will be in one place is the spermatogonium. We'll never find the spermatogonium halfway deep into the cell. Thyroid gland. All right, and this is a hydrocele formed from the peritoneum of the tunica vaginalis again. That's often the easiest way to find a diagram because we don't have any models that show the tunica vaginalis very clearly. So on a lab exam or a quiz, it's usually a hydrocele that I'll show you because that shows it expanded enough to mark.
And on that picture, there's no
Right.
you wouldn't say.
The corona is shown right here. So you would see glands here. All right, back to this structure here. This opening would be the superficial or the deep? Superficial. It's on the external body wall, and deep is on the internal body wall. Told you what this one was already. Primary. All right, which of the following releasing factors stimulates the anterior pituitary gland to synthesize luteinizing hormone, or LH? GnRH, which stands for gonadotropic releasing hormone. Which of those molecules listed up there are made by the hypothalamus? You did the same thing the morning class did. All of them. Notice TRH, CNRH, CRH. They're all
We're separating it out. Yeah.
They come from different nuclei in the hypothalamus. All right. So the first three would come from the ventromedial nucleus and get into the capillary bed through the portal veins to the anterior pituitary gland. Oxytocin, along with ADH, would be made by the cells in the paraventricular and come down the axons to be released from the posterior. But they're all made by the hypothalamus. That's okay, the morning class knows it too. Not that you're in competition with them. All right, so inhibin, this is something we'll talk about in a few minutes. We haven't really talked about it yet. Is along with androgen binding protein made by the Sertoli cells. And like testosterone, it inhibits the pituitary hypothalamic circuit. So I don't expect you to know this one. It's what we're going to talk about right here. Okay? In actuality, inhibin goes directly to the pituitary gland, while testosterone affects the gonadotrophic releasing hormone. Okay? You have this diagram in your lecture notes, which has it reversed. So you might want to draw the testosterone to the GnRH and the inhibin to the anterior pituitary. Okay? It's correct here and wrong here. So let's take a look at that. I didn't leave myself much board space to talk about this. I wanted to take care of this a little earlier, but so we are in male reproductive lecture notes near the end, just prior to the study objectives on the physiology.
72.
So thank you, page 72. So remember we talked about the castrated male and the woman in menopause with low levels of sex hormones. And what happens to GnRH? It's elevated. Okay, so we have that seesaw negative feedback. Just like when we have low levels of cortisol, cortisol releases hormone goes up. When we have low levels of active thyroid hormone, or even inactive thyroid hormone, high levels of inactive thyroid hormone, TRH is going to go up. All right, so it's that seesaw relationship that we have with negative feedback. So I think I'll just erase this part here and use this half of the board. So the hypothalamus under influence of decreased testosterone, okay, is going to increase synthesis of gonadotropic releasing hormone. And how is what organ is gonadotropic releasing hormone going to go to? Anterior pituitary, all right? So GnRH is going to bind to cells of the anterior pituitary. And how does it get there? Does it travel down axons, or does it get there via blood vessels? Via blood vessels. Which capillary bed does it go into first? Cap bed 1. So it's going to go into the first capillary bed of our portal system. And then it's going to enter the pituitary or hypophyseal veins and go down the stalk towards the pituitary gland. So it's going to go into portal veins. And then it's going to go into the anterior pituitary to cap bed 2. And exit cap bed 2 to get to the anterior pituitary cells. All right. There, gonadotropin-releasing hormone is going to bind to its membrane receptor. And the pathway it uses to activate the cells to make LH and FSH is our phospholipase C pt.2 pathway. All right? So I'll just abbreviate that with phospholipase C pt.2. That whole G protein pathway we covered on Tuesday last week. Was it only last week? And then we're going to get IP3 and diacylglycerol and calcium ions affecting the cells to make what 2 hormones? What are the 2 gonadotropic hormones? LH and FSH. All right, so this is going to increase levels of LH and FSH, luteinizing hormone and follicular stimulating hormone. Those of you in the back, can you see down here? Sort of? Okay, I'll erase this and start the top again if everybody has that. So we have hypothalamus releasing gonadotropic releasing hormone, going into cap bed 1, traveling down the portal veins, exiting cap bed 2, and binding to its receptors on cells in the anterior pituitary, activating the G protein pathway Phospholipase C splitting phospholinositol to IP3 and DAG. And those releasing calcium ions and activating protein kinases. Those activating enzymes inside the cells, which make LH and FSH. All right? So now, what vessels do LH and FSH go into? They're leaving the pituitary. So the inferior pituitary or hypophyseal veins. The portal veins, remember, are in the stalk. All right? So can I erase that list there? Everybody has that down? Oh, wait.
And there's no inferior, superior veins within the portal system.
There's anterior, posterior, but that makes it anterior is going to carry away from the anterior, posterior is going to carry away from posterior. Okay? All right. So now we have the anterior pituitary is releasing into the hypophyseal veins. Remember, 2 B's is plural. LH and FSH. Remember, I'm doing abbreviations just to give me more room, but you guys need to be able to write this out. Okay, know what those words are. And those are going to go through these veins to the heart, out to the lungs, back to the heart, and get pumped via the arterial system all over the body. Where in the male, they're going to go into the testes. Okay? And LH is going to bind to receptors on the interstitial cells, the ones that are outside the seminiferous tubules. And FSH is going to bind to the Sertoli cells or sustentacular cells. Are these steroid hormones, LH and FSH, or water soluble? Water soluble. So they're going to use the membrane receptor system again. And this is the cyclic AMP pathway. All right, so they will bind to membrane-bound receptors and utilize the G, they're both G proteins, whatever the one I erased. But this will be adenylate cyclase and cyclic AMP. Okay, that sequence. So the effect of LH as it activates enzymes inside the interstitial cells is to produce what hormones? Testosterone, okay? Our first steroid. So these are making testosterone. So we would expect the interstitial cells to have high levels of lipids, high levels of cholesterol, because that's what's going to get converted into testosterone. And the follicular cells, not the follicular, the Sertoli cells, are going to make inhibin and a molecule known as androgen binding protein. It's not really a protein, it's not even autocrine, because what it does is it just binds testosterone and keeps it in high concentration around the developing sperm cells. Because it's going to go in the blood, and it's going to get carried to the rest of the body, and we want to keep a high amount close to the developing sperm cells. So that's the purpose of the androgen binding protein. The inhibin will get into the blood and bind to receptors in the anterior pituitary. And we have the cycling, not as distinctive as cycling, all right, as we have in the female. But it's about, it takes about 5 weeks for each wave of sperm to be produced. So this slide you have in your lecture notes this slide is in the PowerPoints that are online. Did you get to see those? When you look at content, they're on there as well. Okay. So you can. This is same thing I've just written on the board with the images. All right. So this is a diagram that we'll look at again on Tuesday because it shows the relationship much tighter than I suspected as I was learning this material between male and female. I always thought it didn't make sense as closely intertwined as male and female sequences are. We both make FSH and LH. And LH triggers the male to release testosterone, whereas in the female, it is FSH that triggers the release of estrogen. But, well, it should be the same hormone in both of them because we have a pretty tight corollary between, similar structures. In actuality, it is completely in sync. Again, I'll go over this again on Tuesday. Here is our seminiferous tubule, all right, with our Sertoli cells and our interstitial cells outside of that structure. LH binds to the interstitial cells, FSH to the Sertoli cells, all right? Interstitial cells make the testosterone. Now let's look at the similar structures in the female. The egg is going to be right here in the middle, all right? It's surrounded by follicular cells. That's our egg follicle, our ovarian follicle. And on the outside are what we call theca cells. Don't worry about that now. We'll look at that in more detail. The theca cells bind the LH. The follicular cells bind the FSH, follicle stimulating hormone. That's where we get the name from. So how does this fit? Well, the theca cells take cholesterol and make testosterone, or at least the earlier version of testosterone, called androstenedione, exactly like these cells do. Then, the follicular cells take that androstenedione and make it into estrogen. So we're still following the same sequence, we're just going a few steps chemically farther. All right, so if you look at this diagram here, we'll look at this when we do the female. Here's cholesterol, converted into androstenedione, that's the precursor. And in the male, that's made into testosterone. In the female, the follicular cells will take that and make it into forms of estrogen, or they'll take the testosterone and make it into forms of estrogen in the follicular cells. But this takes place in the theca cells, just like it did in the homologous structures of the male. Okay? So there really isn't a disconnect. They are parallel pathways. Now, one thing I want to mention here, and I'll kind of diagram this on the board as well. So in the interstitial cells, we have cholesterol being converted to androstenedione. You might hear of that molecule in some of our steroid scandals with athletes. Okay, that's a form that's often used. And then this is further converted to testosterone. Most of the testosterone stays as testosterone, okay? And it travels throughout the body and travels to the Sertoli cells. It is converted to an enzyme known as alpha reductase to dihydrotestosterone. It's very similar to testosterone, just a little bit of difference here. Yeah?
Is that's in the Sertoli cell?
That's, yes, that's done in the Sertoli cells. Thank you. And some other tissues, but in the testes, it's done in the Sertoli cells. Dihydro is also known as DHT. This is the enzyme. Remember I talked about that island where some of the kids, I told you guys about that on Tuesday? Where some of the kids don't have enough of this enzyme. And without enough of the dihydrotestosterone, their labia majora, their scrotal sac isn't formed. And they're guys, but they don't have a scrotum and the testes haven't descended yet. And the penis looks like a large clitoris. It's not the penis, the shaft of the penis, the labia minora haven't fused. And all of that normally occurs in the fetus occurs at puberty. So the scrotal sac forms, the testes descend, the clitoris, quote clitoris, develops into a penis when they're around 10 or 11. When they get a surge of testosterone production, and then because of that surge, enough of that gets converted to DHT. So this provides secondary sex characteristics and phenotype. Okay.
What island?
It hasn't been named, it's somewhere in the Caribbean. Now I could use that as a scientific excuse to go, sailing around trying to find it, but they just don't want a lot of people descending curiosity seekers and stuff, but because it's a small area, it's been known since the film that I showed about this, I showed in like 81, 82. So in the mid-60s, they started investigating that. Okay? The rest of the estrogen, estradiol stuff we'll look at on Tuesday next week. Yes?
What does it say after the DHT under that?
Secondary sex characteristics. So the denser muscle, bone, lower voice, facial hair, as well as the genitals looking like a male. Okay, any questions on, again, we'll review the sequencing and do a very similar pathway on Tuesday when we talk about female hormones.
Kind of covered the Sertoli. The Sertoli covers everything from diabetes.
Yeah, so the alpha reductase is inside the Sertoli cells.
Okay.
So when it gets the testosterone,
And testosterone and everything above it.
it's interstitial.
Interstitial.
So it's interstitial cells making that. Okay, scary business now. All the other diagrams on the board. Our various Bio 430 faculty cover the autonomic nervous system to various degrees. We actually had one adjunct about 5 years ago that never touched on it. And that is, in my opinion, the single most, other than maybe cell function, single most important part of Bio 430. I actually added 2 more days so I have 4 days on autonomic nervous system in my Bio 430 class. I added 2 more days last semester. So understanding that none of you were in my class last semester, I'm going to go back and do a little bit of either review or knowledge. If you need more, take a look at your autonomic nervous system early in the textbook, okay? Because all of our systems, cardiac and respiratory and kidney and everything, is governed by neurotransmitters from sympathetic and or parasympathetic components. So, in review, and I'll come back to this in a moment, our central nervous system has nerves coming from both the brainstem and the spinal cord. Okay, if this is familiar, just listen. I kind of want to bring us all up to speed so that what I cover next isn't quite as confusing as it could be. The parasympathetic nervous system is known by location as what? What 2 parts of the central nervous system contribute to parasympathetic nerves? Anybody know? Top and bottom. So this part, the brainstem, is referred to the cranial region because it's in the skull, all right? And the last part of the spinal cord is what part? Sacral. All right? So this is known as the cranial sacral component of the autonomic nervous system. Okay? Well, anybody remember this review part? I'm not going to ask quiz questions or lecture questions on. It's just kind of, again, to bring us all to the same playing field. Anybody remember the 4 cranial nerves that provide the preganglionic fibers for the parasympathetic aspect?
Give us one.
All right. What's the one that causes the pupil of the eye to constrict? Now, optic is vision. Cranial nerve 3. Ocular motor. Okay? So, that's cranial nerve 3, ocular motor. And I don't know if you learned about the ciliary ganglion, but it's the cell bodies of the postganglionic neuron that then go to the iris or to the smooth muscle that changes the shape of the lid, ciliary muscle. Okay? The next 2 go to sweat glands. Not sweat glands. No wonder you're looking at me strange. The next 2 go to salivary glands. Vasopharyngeal goes to the parotid gland. And the other one goes to both the tears, the lacrimal gland, and to submandibular and sublingual salivary glands. Anybody know the cranial nerve that goes to causes tears to be released? It also has motor innervations of facial muscles of expression. So facial. All right. And then the long one is vagus. Vagus means wanderer. And it does autonomic functions in the thoracic and abdominal cavities. All right. So these ganglia are outside the organ that they innervate, the salivary glands. All of the, obviously there's more than one because the vagus has multiple branches, but this ganglion collection of cell bodies outside the central nervous system is found in the walls. So if it's going to the lungs, it'll be within the smooth muscle of the bronchi. If it's going to the heart, it'll be in the wall of the atrium. Going to the stomach, it'll be among the layers of the smooth muscle in the stomach, all right, or near the secretory cells. So that's the cranial portion, all right? I have no idea why we skipped most of the rest of the spinal cord and then start over again down here in the sacral region. But we have preganglionic cells coming out of the spinal cord in the sacral region and going, say, to the bladder, where the postganglionic neuron releases its neurotransmitters, or it may go to the penis or the rectum, okay? Vagus stops essentially at, it goes to pancreas, stomach, kidneys, small intestine, and ascending colon and the transverse colon. So we're looking at vagus a lot as we do respiratory and digestive systems. The descending colon and all the pelvic organs come from the sacral nerves. Okay? So we're going to be looking at the autonomic nervous system when we do the heart, the blood vessels. We'll be looking at it when we do respiratory system and digestive system. Not so much urinary system, though there is, because the kidneys aren't quite as involved, but there's still parasympathetic, except for the bladder that we'll be talking about. Okay, what's the neurotransmitter released by the preganglionic neurons? Anybody know? It's the same as the neurotransmitter released by the sympathetic. Acetylcholine. Okay. So we have acetylcholine as our neurotransmitter. Its effect, its receptors on the postganglionic neuron open sodium channels. All right. So the receptor are sodium ion channels. Always excitatory. What's the neurotransmitter at the organ? Or the effector down here? What's the postganglionic neuron releasing? Still acetylcholine. Okay. So the receptors on the smooth cell or the gland are usually G proteins. They can be excitatory or inhibitory, and we call them muscarinic. These are nicotinic. Skeletal muscle is which one? Skeletal muscle is activated by acetylcholine as well. It's not autonomic, it's voluntary acetylcholine. Just for free, those are nicotinic receptors. Ever heard of the disease myasthenia gravis? That's when your own antibodies block acetylcholine from binding to its receptor, and the person ends up gradually becoming paralyzed and dying of respiratory suppression because they can't use their respiratory muscles unless they're on a ventilator. To overcome that, one of the treatments is to give them lots of acetylcholine. And if too much acetylcholine is given, they can go into a cholinergic crisis. All right? So with a cholinergic crisis, too much acetylcholine, they can have skeletal muscle convulsions because it binds the same acetylcholine. What do you think would happen to their digestive tract? When we have too much acetylcholine, are we affecting only parasympathetic? As you'll see in a few moments, both parasympathetic and sympathetic release acetylcholine here. So when we give someone too much acetylcholine, we can have both sympathetic